Okay, so let's take a brief second and review the cell membrane structure. If you remember, your cell membrane is selectively permeable. Okay, and so what we mean by selectively permeable is that, remember, it only allows certain things to cross back and forth across the cell membrane, and most of that is due to that phospholipid bilayer having that hydrophobic region right there in the middle that kicks out a lot of your kicks out most of your charged particles, anything that's really large. Okay, and we need it to be selectively permeable anyways. That's its job, is to help create a barrier between the outside of the cell and the inside of the cell. Okay, so what we're going to be talking about today is we're going to focus on the different kinds of cell transport. Again, most of this should be reviewed from your freshman year. Things like diffusion, osmosis, active transport. What I need from you guys is for y'all to have a really good handle on this information that you got your freshman year so that you can apply it in different ways in the AP class. Okay, so let's start with regular diffusion then. Okay, so if y'all remember, diffusion is where things go from a high concentration to a low concentration. So they're going with the concentration gradient, and you're down the concentration gradient. This does not require any energy because this is how the molecules naturally want to flow. When we talk about free energy, we'll apply that back to diffusion, right? And we'll talk about entropy and the organization of the system. Okay, but for now, let's just kind of think of it as a crowding situation. Hey, the molecules, as they move around, they like their space. And so when they can spread out into this extra space, they will. So again, high to low concentration, no energy is required, so no ATP is required, and they move with the concentration gradient. Most of our molecules that are able to do this, they're going to have to be small, and they're going to have to be non-polar. Okay. Again, they have to be small and they have to be non-polar. They have to be non-polar so that they can fit across the cell membrane, okay, and they have to so that they don't get kicked out by that phospholipid bilayer. So if you look here, an example of molecules that can do this would be oxygen. And you see your oxygen here, small, non-polar molecule that can diffuse across the cell membrane. You've got a higher concentration outside the cell than you do inside the cell, so it can diffuse across. Okay. Water does not diffuse across. It requires an aquaporin. Okay, carbon dioxide is small enough that it can diffuse across the cell membrane as well. So if y'all remember, in addition to diffusion, we have what's called facilitated diffusion. Remember, it's still a type of diffusion, so this is not going to require any energy. Okay, it's still going to move molecules from a high concentration to a low concentration. Just in this case, the molecules are either too large or they have a charge, and so they need essentially some protection from this hydrophobic region of the phospholipid layer in the middle there. So these are going to require channel proteins here. Like an example there would be your aquaporins, right? They'll provide a channel for that water because the water cannot cross through this hydrophobic region. Another example would be your carrier proteins here. Okay, you can see the carrier protein pick up the molecule, okay, essentially carry it across and release it on the inside. And again, none of this requires any energy. You're still moving these molecules from a high to a low. The molecules just need a little bit of help to get through that phospholipid bilayer. And those of y'all that were in my class, remember we would talk about the river analogy, okay, that we would want it to float down river. It doesn't require any energy to float down river, okay, and as we float down river, okay, no energy, I'm going with the concentration gradient or down river. Okay, that would be our passive diffusion, regular diffusion. Okay, you got some of those people that get in the water and they sink immediately like a stone. Okay, those people they would require a raft or an inner tube. They would be facilitated diffusion. Once they've got that raft or inner tube, once they have that little bit of help, still doesn't require any energy, still going down the river. So they're still going down or with the concentration gradient. So rounding out our types of passive transport, we have osmosis. Hopefully y'all remember osmosis is the diffusion of water. Okay, so it's not going to require any energy. It's a type of diffusion. So no energy, going from high to low concentration. Okay, and so in this case, what happens with osmosis is that the selective permeable membrane, okay, you've got some molecules that cannot get through that membrane, but the water can. And so in these instances, water will move to help even out concentration. Okay, so if we talk about concentration for a little bit, Y'all remember hopefully what concentration is. Okay, concentration is essentially percentage. It's the amount of solute that I have dissolved per unit volume. Okay, so again, it's my amount of solute per unit of volume. Okay, and if y'all remember, solute is what gets dissolved, and our solvent is what's doing the dissolving. So let's talk hyper, hypo, and isotonic for just a second. Okay, so remember we had three basic types of solution. Okay, I've got hypertonic, hypotonic, and isotonic. Can't spell. Okay, and these are going to refer to our concentration of our solution. Okay, so the concentration of my solution. And remember, my solution is made up of two parts, right? Solvent and solute. Okay, so I've got my solvent that is going to do the dissolving, most of the time water in biological systems, and my solute that's going to get dissolved, salt, sugar, whatever it is that's getting dissolved. Those are going to make up my solution. So in a hypertonic, hypotonic, and isotonic solution, you have to compare two things. We're comparing whatever solution is on one side of the membrane to the solution on the other side of the membrane. So when we're talking about something being hypertonic, okay, that means it has more solute than whatever you're talking about. As a general rule in biology, usually we're talking about a cell. So let's say it's got more solute than compared to the cell. Okay, so if I was sitting in a beaker here, okay, and I've got my cell, and I've got 10% solute out here, and 5% solute in my cell, the solution in my beaker would be hypertonic to my cell. Okay, well, hypotonic is going to be the opposite. Remember, hypo is under or less. Okay, so hypotonic is going to have less solute concentration than your cell. And then finally, if y'all remember, iso is the same. Okay, so isotonic solutions are at equilibrium. We've got the same solute concentration on both sides of the membrane. Okay, and remember, those molecules are still moving. Okay, if I've got molecules on this side as well as this side, these molecules are still moving back and forth once they reach equilibrium. Just remember, they're moving back and forth at the same rate. The molecules don't just freeze and stop moving. So let's take a second and talk about how we would determine which direction water is going to move, because you may see some examples of that. Okay, you may see examples of, for instance, let's say I have a beaker and I have a cell, and in this cell I've got 20% solute, and out here I have 85% water. Okay, one of the easiest ways to do this, and I know it takes you one extra step, but it will guarantee you will get this right every single time if you are asked which direction the water moves. Okay, is you draw yourself a little T-chart here. Okay, and so I've got my solution and I've got my cell. Okay, and I've got my water concentration and my solute concentration. Okay, so my water concentration in my solution was given to me. It's 85. Okay, so if my water concentration in my cell, in my solution is 85, then my solute concentration has to be 15, because these have to add up to 100, right? I have to have 100% of my solution. So if 85% of it is water, 15% of it has to be solute. So in my cell, okay, I've got 20% solute, which means I've got 80% water. Okay, so what I have now is I take down here, tells me the name. Okay, so this gives me my name. So if I'm looking for my hypertonic versus my hypotonic, well, 20 is bigger than 15. So my cell is hypertonic to my solution. So my solution is hypotonic. Okay, so my solution is hypotonic, my cell is hypertonic. And I know in the past you've tried to memorize which direction the water moves in both of these. Okay, if you've drawn your chart here, water flows from high to low. Remember, that's what osmosis is. It's a type of diffusion, and in diffusion things flow from high to low. So 85 is bigger than 80. Water goes that way. Water will go into this cell. And we'll talk in just a minute about what would happen to the cell when the water went into it. Okay, let's say you were given a U-tube problem. Okay, so here's my tube. It's a good tube. Okay, and it's got water on each side. Okay, I've got some molecules in there. Okay, and so let's say over here I have a, I don't know, let's make it not super easy numbers. Okay, let's say I have a 17% solute concentration on the right. Okay, and over here, let's say I have a 5%. Okay, very quickly. Okay, I just draw my chart. Solution, cell, water, solute. Okay, so my water concentration. And see, in this case, okay, I wasn't even paying attention. Instead of writing solution versus cell, okay, I'm just going to change this to side A versus side B. And if it's not given to me, I'm just going to assign them. That's A, that's B. Okay, so on side A, I've got 5% solute, which tells me that I have to have 95% water. On side B, I have 17% solute, which tells me I have to have 83% water. Okay, water flows from high to low, right? So water's going high to low. Okay, so water's going from side A to side B. Okay, down here my solute concentration tells me the name. 17 is higher, right? So side B is hypertonic, which means side A has to be hypotonic. Okay, so I've got a hypertonic solution over here. Okay, hypotonic solution over here. Okay, that chart will enable you to get that right every single time. So let's take a look at what these hypertonic, hypotonic, and isotonic solutions do to the different kinds of cells. We're going to look in particular to an animal cell versus a plant cell. And what's our big difference here between the animal cell and the plant cell? Okay, right? Our plant cell is going to have a cell wall, where our animal cell does not. So in a hypotonic solution, when the water is moving into the cell can eventually take on so much water that the cell explodes. Okay, it goes through lysis. Okay, and so if the cell goes through lysis or is lysed, the cell has exploded. Well, in a plant cell, the cell isn't going to explode. It can't explode because it has a cell wall. So it's got this rigid structure, so it's filling up with water. And you remember which organelle in the plant cell is going to fill up with water. That's going to be your central vacuole right here. Okay, and so when a plant cell is totally full of water like that, that's when a plant cell actually looks normal. That's when a plant's able to stand up straight and doesn't look wilty, okay, because at that time it has a very high turgor pressure. Okay, it has a very high turgor pressure. The cell is what is considered turgid. Okay, it's, plants are able to stand upright, and plants don't have bones or anything like that, but what they have is this pressure. And so when the pressure on the inside of the cell wall of the plant, okay, is nice and strong, that makes this plant cell push on the plant cell next to it. So this one's exerting pressure, this one's exerting pressure, and that's what enables the plant to stand upright. Okay, if you've ever played that game where you sit down back to back and you try to stand up without using your hands and you have to push against each other, okay, it's the same concept. So plant cells, plants that look nice and healthy, they are actually in hypotonic solutions. They have a very high turgor pressure at the time. When the plant starts to look wilty, okay, that's going to be your flaccid. Okay, the plant is starting to wilt. Okay, and that means it's in an isotonic solution. For an animal cell, that's when an animal cell is normal. There's equal amounts of water going in and out. Okay, in a plant cell, there's equal amounts of water going in and out, so you don't have this buildup of pressure on the inside. So the plant will start to look a little wilty. Okay. In a hypertonic solution, water is leaving the cell. Okay, and so a plant cell, an animal cell, will start to shrivel, or what is called crenate. You may see this as well. Okay, so the animal cell will shrivel or crenate. Okay, it could eventually shrivel so much that it dies. And a plant cell, okay, will go through what's called plasmolysis, or it'll get plasmolized. Okay, so plasmolysis would be your noun here. Okay, and hopefully you recognize here your lysis on the end. Okay, and so what's happening with plasmolysis is the plant cell is losing so much water that it starts pulling the cell membrane off of the cell wall. That's what's happening here. Is the cell membrane is tearing off of the cell wall. And when that cell membrane is tearing off of the cell wall, that is what's going to kill a plant. So animal cells and plant cells respond differently to the movement of water. So now we have the opposite of our passive transports. We have active transport. So active transport is going to require ATP. It's going to require energy. And in active transport, molecules are going to move against the concentration gradient. So they're going to require proteins and ATP. Okay, they're going against the concentration gradient. So in this case, they're going from low to high. Okay, for example, on this one here, you have sodium. And we're going to look this example in depth in just a minute. But you have sodium that's being pumped across the membrane, which is going against its concentration gradient. And you can see your concentration gradient over here on the side. Same with potassium as it gets pumped into the cell. Okay, and so that's going to require energy, okay, and it's going to require proteins to be able to do this. So let's look at two examples a little bit more in depth. Okay, so let's start up here, okay, with our sodium-potassium pump example. Okay, so this is the sodium. That's a plus sign up there. These are both plus signs. I can't write tonight. There's a sodium-potassium pump. Okay, this is very, very important in our bodies, in particular with nerve conduction. Okay, so we've got a sodium-potassium pump, and we're going to use these proteins, which are also sometimes called permeases, because they are able to permeate the membrane or pass through or across the membrane. They're also a transport protein. So what we have here is we start with the sodium, which is on the inside of the cell here, and it's going to bind to this transport protein, to this permease. So the sodium binds to the transport protein, and then here's where the ATP comes into play. The ATP phosphorylates. When we phosphorylate something, okay, let me spell that. Oh, it's been written for you. Yay. So when we phosphorylate something, we just add a phosphate to it. And so when this protein here gets phosphorylated, it changes its conformation or its shape. And the change in the conformation or the shape essentially closes off this end and opens up the end on the extracellular side. So now that sodium gets spit out of the cell membrane. And in this conformation, that allows potassium to be able to bind to the transport, to the permease here. And this permease will then take up that potassium, okay, and it's going to release this phosphate. And so when it releases the phosphate, it goes back to its original conformation, which is open up into the cytoplasm here. So now the potassium can get put into the cytoplasm. And so this will continue to pump. This does have a purpose. Okay, it helps create a concentration gradient. In particular, in our nerve cells, we need this. We need sodium to be piled up on the outside of the cell so that it can quickly flow back in to help promote nerve conduction. Okay, so let's look at another example of active transport. So our second example here with active transport is a proton pump. It is also sometimes called an electrogenic pump. Okay, so this electrogenic pump, that's a C. Okay, so it's also sometimes called an electrogenic pump. So an electrogenic pump or a proton pump is going to pump ions, in particular usually hydrogen ions, against the concentration and charge gradient. Okay, you'll notice the hydrogen ion, it's got our positive charge here, and the inside of the cell is a little bit negative. Okay, so it's forcing this hydrogen ion with more positives, so you know positives don't like each other, they repel, as well as against the concentration gradient. So I've got lots of hydrogens out here already. So now I'm going doubly hard here, against the concentration gradient as well as against the charge gradient. So this is going to require some ATP to force that hydrogen across the membrane there. Okay? And what will happen is this will separate the charge across the membrane. It'll make the inside distinctively negative, the outside distinctively positive. Okay? And also it can sometimes be coupled with another protein, like you see down here with this sucrose, okay, that will do a different function. It facilitates diffusion of something else. For example, here as this hydrogen is pumped out and then comes back in here, okay, that also helps bring sucrose back across the cell. You need that hydrogen to be able to bring the sucrose, bring the sugar into the cell. These hydrogen pumps, these proton pumps, will be extremely important when we do cellular respiration and photosynthesis. They create the gradients that really help drive those chemical reactions. Let's take a quick look here at our endocytosis. Remember, endo, we're taking something into here. Cyto is cell. So I'm taking something into the cell here. So endocytosis, I'm going to move large particles into the cell. I'm going to use vesicles to do this. Remember we talked about on the cell membrane video, it talked about how the cell membrane can change sizes. It can change size and shape due to how it interacts with the vesicles. When it's releasing a lot of things, the cell membrane would get larger as those vesicles blend in with it. If it's taking in a lot of things, the cell membrane would get smaller as it forms these vesicles. Okay. So endocytosis, we're taking things into the cell here. Okay, I've got three basic types that we're looking at. I've got phagocytosis, which is also sometimes called cell eating. Okay. And then I've got penocytosis, which is sometimes called cell drinking. And then we have a new one that you guys did not learn about last year, receptor-mediated endocytosis. So phagocytosis, remember, your cell eating. You're taking in larger, more specific particles. Things like amoebas will do this when they eat. Okay, your white blood cells, your macrophages in particular, will do this when they find something that they need to get rid of. Okay, so they're looking for larger particles, that they're looking for specific things. Okay, and if essentially the cell is going to reach out, grab it, okay, and then pinch off that vesicle. These will seal closed. Okay, so these will seal closed here. Okay, and so it will pinch off into the vesicle. Okay, with our penocytosis, it is not as specific. Essentially, with penocytosis, the vesicle is formed here, and whatever falls into that vesicle fell into that vesicle, and that's what the cell gets. So it's not nearly as specific. Receptor-mediated endocytosis is very specific. Okay, this is looking for very particular things here. Okay, and so what happens with receptor-mediated endocytosis is you have these particular receptors on the cell membrane, and when the substrate binds to those receptors, that will start phagocytosis. Okay, so that will trigger the process of phagocytosis. But it has to happen when a specific substance is binding to those receptors. So again, like I said, it's a lot more specific. So last we have exocytosis. Remember, exo is taking things out of the cell. Okay, exo is out, cyto is cell. So here we're spitting things out of the cell. So if you look here, remember your endomembranous system. I've got my Golgi apparatus here. Okay, and so my Golgi apparatus has these modified proteins. Okay, and these modified proteins are now in their vesicles, and their vesicles are traveling over to the cell membrane, and those vesicles will fuse with the cell membrane, and they will spit out their contents. Okay, so exocytosis happens a lot in cells that make secretions for multicellular organisms that need to go to different places. So for a quick summary, before y'all come back to class, I would like you to copy this chart and go ahead and get this chart filled in. Okay, just check off what's going on with things, list your examples. Okay, but just kind of go ahead and get this summary chart filled in.